Hello, good afternoon, everyone, and welcome to this third session of SFAF 2022. This is teaser number 2, and if you're just joining us, you are joining into the NGS panel session, where we're going to hear some perspectives on the pandemic and some of the tools being used by the NGS technology companies, against the pandemic, and also hopefully hear a little bit about the latest and greatest advances in sequencing technologies. So today, the session is being chaired by myself. I'm Sean Starkenberg. I'm a scientist on the Applied Genomics team at Los Alamos National Laboratory, and I'm also the Deputy Group Leader for Biosecurity and Public Health in the Bioscience Division. I am joined today by Bob Fulton. His picture is up there in the corner, a younger version of Bob, but he's still doing quite well. Thank you very much. I'll say that for him. Today, we have 2 speakers for you, Jason Smith from Illumina and Meredith Ashby. And just a little bit of the logistics for the session. Please put your questions for either speaker in the Q&A box, not the chat. And then you can direct those questions to either at PacBio or at Illumina or both, and depending on the flavor and type of the question. And then we'll capture those questions, and then the speakers will answer those questions after both presentations are complete at the end of the session. So without further ado, I want to introduce Jason Smith, who's the Senior Sales Specialist in the Microbiology Infectious Disease Division at Illumina. Jason has been doing genomics for over 20 years, dating back to the Human Genome Project. In the past, he's also worked in the National Lab System in the Pathogen Informatics Genomics Group at Lawrence Livermore National Laboratory, where he provided multidisciplinary expertise for post-9/11 pathogen surveillance within the Department of Homeland Security. And after working with the National Lab System, Jason moved into industry to help drive awareness and adoption of next-generation sequencing. Jason is currently an infectious disease and microbiology specialist with Illumina, and he's focused on the utilization of next-generation sequencing for applied microbiology, research, and infectious disease testing. At Illumina, Jason also acts as a subject matter expert for end users interested in implementation of microbiology and infectious disease testing. So with that, I'll turn it over to you, Jason.
Great. Thank you, Sean. Thank you all for joining today. I appreciate the time and attention. So I wanted to put together a presentation here that gives a little bit of a peek into where we've been. We need to know where we've been to know where we're going. And the idea here is to have a quick, brief retrospective on the SARS-CoV-2 pandemic, and then also looking out broader pathogen surveillance with the Illumina tools and next generation sequencing in general. We'll talk about the applications itself and some of the most recent publications on this as well. So we heard from the panelists previously, and I think we can all agree there's been an unprecedented amount of sequencing and overall pandemic awareness in terms of the data aggregations that we can get. These are worldwide efforts that are happening, and aggregating that data is going to be key. I do want to call out in terms of GISAID's capabilities. They originally were set out in terms of the EpiFlu, but it's been utilized here for SARS-CoV-2 as well. There was a base app provided by Illumina that actually gives you the ability to go ahead and directly submit your sequences directly up to this database. And it was something that Illumina put a lot of resources behind to make sure that we can enable the community to aggregate this data. And as a result of that, back in April of last year, over 80% of the sequencing data within GISAID was generated on Illumina sequencers. If we think about what has actually happened in terms of the variants, the rise and fall, we look at this in terms of a timeline, which you see on the right-hand side. We know that there is a growing risk in terms of RNA viruses to mutate, and we see that within the population in terms of fitness. So we saw with the rise of the Delta variant here over the Alpha that the amplicon-based technologies did suffer somewhat. And that's natural in terms of how you're designing these molecular assays to actually detect them. So the Arctic Consortium wound up putting together a new complete redesign for this, which they're calling the V4 primer pool. And so this is something that can be implemented.
Jason, sorry to jump in here, but I don't think we're seeing your slide deck.
So just to recapitulate, this is the only slide we missed here. In terms of data aggregation, it's a good visual here in terms of the depth of the data that's actually required to get the phylogeny across these genomes. And we know that they mutate very rapidly. And it's also been a global effort in that regard as well. So as mentioned here, the ability of the RNA viruses to mutate will then subject these molecular assays to essentially have potential gaps in their ability to continue to detect these variants. And we see this on the right-hand side with the rise of Delta happening around April of 2021, and then also Omicron coming in and giving another monkey wrench into our capabilities to actually surveil and get full genome coverage for this. The Arctic Consortia here did a complete redesign of the V4 primer pool. And the reason why I bring this up is that in terms of the primers themselves, they can actually be implemented very quickly in terms of a swap out for utilizing the COVID-seq assay by Illumina, which is an amplicon-based assay, to go ahead and give 99% or better coverage of the SARS-CoV-2 genome. What you're seeing on the left-hand side is the V3 primer pool run against the same samples on the right-hand side for the V4. And we know that the V4 primer pool actually addresses the spike protein differentiation that we saw between the samples, and what you're seeing on the top versus the bottom are the technical replicates. So there's good reproducibility within this data as well. As I mentioned, the story doesn't stop with Delta. Omicron came along and gave us another loop here in terms of the capabilities to actually utilize the consortia's Arctic V4.1 primer design. There was a recent app note that was put out by Illumina in order to enable this as well. We provide the spike in pool primers themselves along with the formulation for it. And then this gives even better detection across the spectrum of variants that we would expect to see. So if we just take a quick look into this in terms of what we would expect, you see across the top here, the V4 sample, you do see amplicon dropouts for the SARS-CoV-2 genome for Omicron's sake. But with the V4.1 spike in, we're able to go ahead and rescue most of these regions. And what you're seeing across the bottom here is not just which regions were adversely affected, but in terms of being able to see the regions that we expect to see performance maintain its capability and proficiency. We see that on the left-hand side of this bottom graphic highlighted in red. But more specifically on the right-hand side, you can see the red and yellow arrows here are rescued amplicons that come as a result of this. So this all leans back to the idea in terms of amplicon sequencing being a nice tool for genomic surveillance and the ability to sequence the entire genomes for things like SARS-CoV-2. But there is going to be an effort that's continually had in terms of being able to make sure the efficacy of these assays are maintained. So another thing that I want to bring up in terms of my background, which Sean mentioned in there, working for the pathogen informatics group, the rubber really hits the road, and not just data generation, but data interpretation. Essentially, what can you do with this information that gives you very quick, actionable results? And more specifically here, the ability to actually characterize this information in a way that's usable. So I want to call out that we have the Dragon COVID Lineage App. In the early days of COVID-seq, there was a COVID-seq test pipeline, which combined part of what you're seeing here on the screen. But the take-home message from this is now that you have a very simple capability within BaseSpace to be able to take raw FASTQ reads, place them directly into an app on BaseSpace, generate the FASTA sequence that's required for submission to GISAID, but it also gives you the lineage and clade information as well. So we're pulling together these pieces to make this a more formidable approach in terms of data analysis capability for the field. And then we also maintain updates on this as well in terms of the Pangolin and XClade databases. So we start to think now in terms of broader surveillances, in terms of pathogens with some of the Illumina tools that we have out here. I'd like to cover at least in briefly here, the idea of the Respiratory Viral Oligo Panel. So beyond SARS-CoV-2, there's a capability using hybridization capture on these libraries to actually go ahead and sequence greater than 40 viruses in a particular panel. Now, in terms of a full metagenomics approach, that's certainly viable as well. But the question typically comes up in terms of, I'm looking specifically for these particular organisms or these viruses of interest. How can I actually pull those out of solution and essentially sequence those and get full coverage of those genomes? So the way that this happens, to give you a brief overview, or I should say a high-level overview of the workflow, is that we go through the cDNA conversion. We then tagment and create the libraries, and then post-library creation, the capture is actually performed. Now, we've allowed for a modular configuration of this to allow for some cost efficiencies there. In terms of quant and normalization here and going onto the sequencer, this is a really smooth workflow in order to actually go ahead and target your viruses of interest for this particular panel. This is not meant to be an eye chart, it's just to give you a capability to dive down a little bit in terms of the content for these particular panels. But I do want to call out again that this is full genome coverage for all 40 plus of these viruses. And that's what the probes are designed against. They're able to actually go ahead and pull out full coverages of the genome for these in a single assay. And it also allows you to essentially do this in a precision metagenomics approach for this. We do include probes for positive controls for human as well. And there's actually been a redesign of this to give even more high sensitivity for the viruses in a V2 version of this panel. Continuing on in the idea that once you have that information, the next question is, what do we do with it and how do we interpret this information? We do provide a application on BaseSpace as well called the Dragon RNA Pathogen Detection App, which is specifically designed to go ahead and target the organisms that are listed in that panel for RVOP. And in the ways that you would expect from the COVID Lineage app and some of the other BaseSpace apps in terms of the microbial space, we actually provide you with a tabular format that's downloadable. It gives you not only the detection, positive or negative, but the coverage that you would expect from that particular genome as well. If we want to broaden the scope even further, what if respiratory viral pathogens are not the only interest, which I would imagine is part of this group as well? We can expand that out to multiple kingdom as well. Now, in terms of bacteria and the fungi that are present here, but also the AMR targets, this is something that we're really excited about. And in terms of the panel itself, it runs on the same exact backbone as the Illumina RNA with enrichment protocol, which I highlighted previously. So the idea of essentially generating a workflow for this would be exactly the same that you would see from RVOP. It's just changing the content essentially of what you're targeting within these particular panels. To give you another brief highlight here, we do have full genome coverage of the SARS-CoV-2 and influenza viruses in this panel. But you can see here, there's a number of different other agents in here in terms of interest. What you're seeing right next to the names are the different versions of that particular organism. But I also want to call out here that we do have AMR targets as well, along with their associated organisms. And typically the next question that comes from this is, Jason, that's great that we can detect all these, but again, what do we do with that information? So in the spirit of having a full solution available for this, the Rpip panel, the Respiratory Pathogen ID and AMR panel, which we refer to as Rpip, has a BaseSpace app that's available as well, and we call that powered by Expify. This is the platform that is enabled with our partnership with ID by DNA. This provides you with a number of different data formats, but in particular, you're seeing the PDF of the report that's available here. I do want to call out, it not only gives you detection, but it also gives you quantification capabilities with some built-in controls that are available there as well. And then it will go ahead and aggregate all the information that we have in terms of the targets that are expected across those multiple kingdoms, and then being also to provide the AMR targets that were detected as well. All right, with the idea of the tools that we have in terms of the updates there, we always want to think about what is the next step. So we've lived through these harrowing years here in terms of understanding what the coronavirus SARS-CoV-2 is doing within the population. There's always the diagnostic side of things, but in terms of using other tools like this for environmental and clinical monitoring, I do want to highlight this for a couple of different pieces that you may not be aware of since the last time we met. So there was recently an app note that was put out by Illumina in terms of wastewater surveillance, utilizing a couple of tools I just highlighted. So first and foremost, this is utilizing samples that came from BioBot in terms of wastewater collection sites. We took the COVID-seq assay along with the V4 primer set, along with the respiratory virus oligo panel V2. We compared that against these samples. I want to highlight a couple of the findings. Just to give a little bit of orientation here, the samples that were collected from the wastewater, some of the samples came from the spring of 2021, and some of the samples came from fall of 2021, which I'll show you in the highlight here. So if we zoom in on COVID-seq, what we're able to do from these wastewater samples was go ahead and detect the difference in variances that we would expect to see at those particular timeframes, which matches up directly with the historical knowledge now about what was happening at those times. So you see here on the left-hand side, alpha was dominating in the spring of 2021, and delta quickly rose up, But this gives you the capability to actually see essentially a profile, if you will, in terms of what the actual variants are doing within these wastewater samples. And it's been shown that the wastewater samples are typically a leading indicator because you are in a population-based survey, if you will, in terms of the multiple variants that could be circulating within a geographic region and a large base of different sample sources. I do want to call out that the data analysis for this particular case was utilizing Callisto, which gives you the capability to actually see these different variants that are present. Switching now to a more broad view of this, the same samples were taken and one run with the respiratory virus oligo panel, RVOP. What you're seeing from left-hand to right-hand side here is essentially different CT values for those particular samples underneath the bar graph or the pie graphs. But you can see with a higher CT value for SARS-CoV-2, which would indicate lower amounts of SARS-CoV-2, you do see that there is a population of pathogens that are present as well. So RVOP is a really nice tool to be able to essentially survey these and essentially detect which pathogens are actually present. You can see on the right-hand side here, these line up directly with some of the key organisms that were highlighted previously within the RVOP pathogen the RVOP probe set that's available. The other thing to think about here in terms of viral prevalence, the next thing to really dig in here would be essentially this 45% you see on the left-hand side. Can we do variant detection for that? And that's some of the work that we're currently doing as well. So in addition to the tools that are available, I do want to call out that there are programs that have been set up actually early on in the pandemic in terms of national wastewater surveillance. So on the left-hand side here, you see the FDA has a genome tracker group that's available for this, and the sequencing submissions are coming in. On the right-hand side, the CDC has a national wastewater surveillance system as well. So this is something that in terms of interest in signing up for that, it's available here on the websites themselves, but there's some really interesting data that's coming out, not only in detection of presence and absence, but essentially what is the wastewater aggregation across multiple genomic regions, which we're finding is going to be a very useful tool and informative tool in terms of our capability to surveil for pathogens. And we're able to get into that next phase here in terms of what we've done previously in the pandemic with the tools in our toolkit. So to push this even further, I want to highlight a recent paper that came out from the Medical College of Georgia. They were looking at next generation sequencing, specifically with RVOP, to go ahead and address some of the public health questions that come out. It's not just a question of which ones are present, but transmissibility within the region as well. And that's exactly what they did with this paper. So just to call out what was happening up front with the data generation here, they ran RVOP on a NextSeq at a 2x150 read length, and they utilized the Dragon RNA pathogen the RNA pathogen detection app to generate the information. They also used the Metagenomics app as well. Some interesting results that they found. The co-infections were present with SARS-CoV-2. I think that's been discussed at length, both in the media and within these circles as well, that co-infections is something that's of keen interest here, not just seeing what's positive for SARS-CoV-2, but understanding what other sort of viruses may be transmitting along the way. I also want to call out that there were other negative samples that came in terms of SARS-CoV-2 that highlighted positive for things like human parainfluenza virus 4A. So this gives you an idea in terms of what other co-circulating viruses may be in play as well. And so you're seeing that here in terms of the image itself. They get the phylogeny, and then they also get the transmissibility in terms of those spheres representing essentially the density of those particular samples. For the paper, I do want to call out that they were able to obtain more than 90% coverage for the SARS-CoV-2 genomes, which they originally set out to do. And that is enough to give you this phylogenetic capability in order to generate these sort of images and tracking and transmissibility conclusions from this. They also compared this to qPCR. I'll leave that into the paper as an exercise, but essentially they were seeing that there was very favorable positive percentage agreement along these other metrics and high overall accuracy of the panel and utilization for this as a public health tool. So some current events. In this last week, it's actually come out that the Deltacron variant has been confirmed and was recently submitted to GISAID. This data is now available online, and we're currently looking at Illumina as well. In terms here of the quote here from the technical lead for COVID-19 for WHO, it's critical that we continue sequencing, and we at Illumina are committed to this idea in terms of enabling the field to be able to do this. And in terms of what's been put in place for surveillance, I think our panelist, and Duncan specifically was talking about this earlier, is that we've got essentially an infrastructure now which needs to continue to be built upon and allow us to push out and get into this next phase of surveillance, because what's now in the headlines has a high level of capability to be seen across the world. And it's something that we're in a unique position to be able to push forward on the technical precipice of what we're actually accomplishing right now. And just as a final note here, Illumina is committed to this in a number of different ways, not just within the US, but globally. So Francis is committed to donating $60 million into this global pathogen genomics initiative, which is actually building upon the African pathogen genomics initiative. And this involves not only the platforms, but also the reagents and the training and the support that is necessary for this in order to push it into the next phase. And so we're really excited at Illumina in terms of what the capabilities are for sequencing to be able to get us into a phase where now we've lived through the pandemic of this age. We haven't previously been into a pandemic state before where we've had these types of tools available. So now that we know what our tools are, we know the enemy that we're fighting and how to actually assay it and understand it even better, we can then implement that in actionable information around the world and specifically utilize sequencing in order to better the lives of everyone in terms of the global network as well. So with that, Sean, or maybe Bob, I can turn it back over.
All right. Well, thank you, Jason. That was great. And so we're going to immediately transition over to Meredith. And again, just a reminder to put your questions in the Q&A box, not the chat box, but the Q&A, and we'll address, and if you could address those specifically to either speaker or both, we'd appreciate it. Then without further ado, next up we have Meredith Ashby, who's the Director in Market Strategy for Microbial and Cancer Genomics at Pacific Biosciences. Dr. Ashby has been with the PAC Bio since 2009 and has worked in R&D on both sequencing, optimization, and bioinformatics applications development. She is now focused on facilitating the use of PAC Bio long-read sequencing to solve the unmet need of scientists seeking to understand the incredible diverse range of microbes that live in, on, and around us and the systems we depend on. So with that, take it away, Meredith.
Thanks, Bob. And thanks, Jason, for that really interesting talk. You know, I think your closing comments in particular about, and the quote regarding the need to continue sequencing really resonated with me and aligns with the thinking of PAC Bio as well. You know, one of the outcomes of this pandemic has been that there's been all kinds of infusion of funding and equipment and resources into our public health entities to upgrade the sequencing by surveillance capabilities that they have. And we at PAC Bio are excited to think about what that might mean for surveillance of pathogens beyond COVID moving forward. And so that's what I'm going to talk to you a little bit about today is using HiFi sequencing, not just for COVID surveillance, but also looking forward to bacterial surveillance and using metagenomics as well to do culture-free methods of surveillance. So just very briefly, you know, PAC Bio uses HiFi reads to do sequencing, and they have the unique quality of both being very long and highly accurate, right? So PAC Bio HiFi reads here have a minimum accuracy of 99.9%, which really, you know, empowers us to do some unique things with sequencing. So the first application I wanted to talk about is our HiFi Viral SARS-CoV-2 kit. At the fall first preview for SFAF, I let you all know that this kit was on its way. Well, it is now out, and I can give a more complete description of what it entails. So it uses a unique approach compared to a lot of the other COVID sequencing solutions that are out there, namely molecular inversion probes. And MIPS are interesting in that they kind of combine the strengths of target captures, meaning they can be deeply tiled in one single reaction pod, but with the low cost and the simplicity of a PCR-based approach. So the way they work is that you have a probe. The probe has two arms, and the arms sit down on the target, and the probes are connected by a DNA backbone. And during the sequencing reaction, what happens is that you basically make circular molecules, which can then be enriched away from the template and other non-captured DNA and turned into SMRT bells and sequenced on the Sequel IIe system. And one of the real strengths of this approach is because we have very deep tiling, 22-fold coverage of any given base in probes, this method is very resilient to mutation-induced dropout. So anybody who's been following the pandemic has seen that the Arctic primers have had to been revised several times. We're now on V4.1 with Omicron. And what we found is that with this kit, we have not had to do any probe updates so far. We get the same robust performance, whether it is samples that are primarily alpha earlier in the pandemic or delta over the course of last fall, or Omicron samples more recently. And the kit also is another strength that it's very simple to use. It's add-only workflow. There's a color change, which each of the three reagents that you add. And so it's very simple. And it also doesn't use that much plastics as compared to a lot of PCR approaches. And as we all know, during this pandemic, one of the pain points really has been labor shortages and, of course, supply chain problems, including for plastics. So our customers who are using this have told us that that is a really helpful feature. In addition, there is automation available for this, but if you don't have an automation system in your lab, it's very easy to use and can be done easily without any errors by staff. And then finally, it has flexible batch sizing, so you can run as few as 24 samples or up to 384 in a single run. So it's great whether you're in a surge or in a lull. So that's COVID, but I think everywhere in the country now we are enjoying the lull here. We are kind of past the Omicron wave in the US at least. And hopefully that will continue. And so we have started thinking about what are the next applications that we should be enabling and standing up for public health now that we have these tremendous more resources and the attention of funding bodies. And one of those is, of course, microbial whole genome sequencing. I think the COVID pandemic has really shown us the utility of having real-time, high quality, complete genomic information about pathogens. And there's an opportunity to kind of upgrade what we're doing for bacterial surveillance as well. So, we have we have during the pandemic at PAC Bio been making some pretty substantial investments in all of our pathogen sequencing applications. So in the case of HiFi microbe, we have migrated the whole pipeline over to HiFi sequencing, and we have increased the number of samples you can sequence in one run to 96, so doubled it. And we're still maintaining the same single contig chromosomes at a price that's very competitive with short read draft genomes. So again, we have migrated to HiFi, but it is the same closed genomes that you are used to getting with PacBio assemblies. And so here we have an example of 96 SmartBell libraries that were made from 24 distinct bacteria that are of interest to public health folks. And they encompass a range of GC content, genome size, and assembly complexity. And what you'll see is that this is without any manual curation at all. It runs automatically. This is outputted. We are getting nearly all assemblies with closed chromosomes. And I know for a fact, because I did it myself, the two that did not close there, if you tweak one setting, that one will close as well. And of course, this is done at very high accuracy because HiFi data really is setting the standard for accuracy these days in sequencing. And of course, with long read sequencing, as a bonus, you get almost all the plasmids as well, which is really important when doing things like looking for AMR genes, because that's where a lot of the action is happening is in the plasmids. So the last application I wanted to talk about is metagenomics. So in this area, there's been a lot of effort, both internally and by scientists working organically with our data to optimize the analysis pipeline specifically for HiFi data. And it's really improving the resolution power, both for taxonomic profiling, so looking at what species are present, but also assembly as well. So in the case of taxonomic profiling, there was a really terrific article that came out recently by Dan Pardick, who is a scientist at PAC Bio, and also Titus Brown and Tessa Ward at UC Davis. And they looked at a number of different data types, and they ran them through a range of different pipelines, which you can see listed on the bottom right there. Some of the pipelines are optimized for short read methods, and some of them are optimized for long read methods. And the take-home message here really is that when you do this work of optimizing or retooling pipelines to really leverage both the long read length and high accuracy for HiFi, you get tremendously improved results. And so these pipelines are now available on the PacBio GitHub site. And what we're seeing is that precision and recall are up tremendously when you use the fully optimized pipelines that are more and more being developed for this data type. And with those more optimized pipelines, the bonus is that you can now run more samples on one smart cell, right? The data is being used much more efficiently and robustly. And as a result, whereas previously we would suggest only running 8 samples on a smart cell, now we recommend that you can multiplex up to 48 samples in one run if the goal is to do taxonomic profiling. But HiFi reads are also very well suited to metagenome assembly. So last fall, I showed this slide, which really shows how metagenome assembly is unique and really more challenging than a lot of other kinds of assembly. And the reason is because metagenomes will often contain very closely related strains. And normally we consider a 3% difference being the cutoff for a distinct species. And so there are things that are even more similar than that in typical metagenome samples. So if you're using noisy long reads, the challenge there is that they first have to undergo an error correction step before they can be assembled. And what can happen is that during that error correction step, things that are low abundance often will be washed away or averaged out. And having erased those differences, you end up with more tangled assemblies or missing species. Whereas with HiFi reads, there is no discrete error correction step. You go straight into assembly. And as a result, you can really see all the different strains. Things that are real don't look like errors, basically. So now there have been some publications which really put some evidence behind that claim. So this is a graph from a paper that was published by Derek Bickhart and his team at the UCSD, and also Pavel Pesner. And I'm throwing up an assembly graph here because I know the SFAF audience tends to have a lot of bioinformaticians in the audience. And so I was confident you all would be able to follow along with me. So the idea here is that the data was assembled both as HiFi data, but then also kind of taken apart and assembled as the single pass subreads, which go in to create the HiFi data. And in this way, you could do a comparison between noisy long read assembly and a highly accurate long read assembly. And so what was done in this case is that they took the bins that were generated from HiFi assemblies, and then they mapped the contigs from those bins back onto the assembly subgraph. And on the right, what you can see is all kinds of bubbles that are reflective of kind of the loss of resolution in that CLR assembly bin, right, which is bubbling up when you overlay them with the more highly accurate HiFi reads. And the consequence of that is that in a noisy long read assembly, those will collapse and appear only in one bin. But on the left, you can see that there are fewer ambiguous overlaps in this layout step, which really enables, in fact, lineage resolution. And that was the highlight of this paper, is that they were getting strain level lineage resolution from the HiFi data. There was another paper that came out recently on HiFi ASM meta. And so this is a pipeline that really is optimized both for highly accurate long reads, but also specifically for the challenges of metagenome assembly. And so in this paper, they basically pulled in all of the data sets that they could find that were public, including the data set from Derek and Pavel Pesner here, but also some human data sets that we have made available for exactly this kind of activity. And the takeaway here is that, these are all great assembly tools, but Metafly was really developed for noisy long reads, and HiCanoe is optimized for single species assembly. And when you retool the back end or develop a tool that really is optimized to take advantage of both the data quality and has baked in the right assumptions about the sample that you're working with, that's when you get the best results. So you can see across the board for all of these different data sets, you're really getting the most complete and the most high quality mags that you possibly can when you're using HiFi ASM meta. And so we have adapted all of our analysis pipelines on the PAC Bio GitHub website to make use of that. And then finally, there's been one further enhancement to the mag assembly pipelines, and that is the addition of circular awareness to the binning step for the HiFi mag pipeline. So what our bioinformatician, Dan Portick, realized was that when you go and do the binning, it operates under certain assumptions, and it did not have within it the knowledge that if you have a closed circular chromosome, that is an intact, complete genome, and that no other things should be added to that bin. And so when he added that piece of information, what he found is that the yield of high quality mags went sharply up, right? And so this is what I mean by, pipeline development and optimization. There is so much more juice that you can get from the squeeze when you're using the right tools for the job. So I just want to close with sharing a couple quotes from customers, which I hope reflect our enthusiasm at PAC Bio, that we really are trying to set a new standard for pathogen surveillance applications, be that virus sequencing or microbial whole genome assembly or metagenome assembly. And we hope that you will keep an eye on what we're doing and download some data and give it a try yourselves. Thanks.
All right. Thank you, Meredith. For whatever reason, I can't start my video, but that's probably a gift to everybody.
Nonsense.
There we go. Sorry, all. Okay, well, great. Thank you, Meredith and Jason, both for great talks and sort of leading us into further discussion here. So in the Q&A box, which I think you should both be able to see, if you guys want to work through some of these questions, and we'll continue to feed additional ones as they come through.
Yeah, absolutely. Meredith, I think the first one is directed at PAC Bio for a HiFi run. Can you see that question?
Yes. So how many COVID samples can be multiplexed in a HiFi run? So up to 384 samples per run.
And Meredith, if I can sort of expand on that. So is that a single barcode in the MIP that you're reading to identify the individual samples? And could you expand on that with, is it a sequencing capacity level, or is it presumably it's more of a complexity of indexes?
So it's so the way it works is that the MIP selection doesn't have the barcode in it. So you do that capture step in every well of your plate, and then we have a second plate which has forward and reverse barcodes. So the MIPS, when that's done, it generates basically a circle, right? And then to make the amplicon that you're going to sequence, you add primers, right, which bind to those probe, the backbone, and add a barcode to it. Right. And so that's where the barcoding happens. And when that copy gets made, that results in a linear piece. And there's a really great video, which is much, much better than my hand signals on our website, but hopefully that kind of got the point across. Yeah, and we went with 384 because, we asked around customers and we didn't see that there was really much need to go higher than that. And also 384 samples results in adequate good coverage on the platform in one run. So.
And what's the movie length to resolve those and ballpark quality?
So let's see, it's the general HiFi quality that you would always get. So most of the data is, all the data is at least 99.9% accurate. And in terms of the movie length, I believe it's a 10-hour run. And hopefully my product marketer, Sarah King, is not going to call me out. But I do believe it's a 10-hour run because it's a relatively short amplicon.
Got it. All right, Jason, you want to take the next one or start with the next one? Looks like it's directed to both.
Yeah, absolutely. I'd be happy to lead off. So the question is, the keynote speakers highlighted the need to detect multiple variants in the same sample. Are you all planning to create more targeted products to track co-infections with different strains? So just inferring from that question, it could be SARS-CoV-2 related or co-infections with other pathogens as well. I think it was actually highlighted in the wastewater paper, knowing that that's surveillance of, say, an environmental sample, but the same thing could be done for clinical approaches as well. And that's really what the paper from the Medical College of Georgia highlighted, is you have the capability to not only utilize, say, a COVID-seq type assay to detect multiple variants, and essentially the rubber hits the road in the analysis, your capability of your databases to compare against them in order to detect them, that's available today. And then looking at it more broadly in terms of other pathogens, absolutely, that's what the hybrid-based capture is aiming to do, is to allow you to detect not only co-infections, but also co-circulating viruses within the same population of samples as well.
Yeah, on the PAC Bio side, so the HiFi Viral Kit is using a 675 base pair amplicon. And the reason we went with something a little longer is specifically so that we could do some level of phasing And in fact, the pipeline that the data runs through for the HiFi Viral application will flag samples where it appears that there's more than one COVID strain in that sample. And again, in terms of other viruses, this is very well known quality of PacBio data, which is that you can sequence as long an amplicon as you want for any kind of virus. And because of the high accuracy, you can basically get to complete quasi-species resolution, where you can see every variant of HIV, for example, that's in a patient. There was a really great article that came out, I think, last year, doing single-cell sequencing for influenza using HiFi, and showing that even within one single cell, you could see mutations in the influenza virus. Nasty little bug, that influenza.
Hard to believe we can't get that vaccine right.
Yeah, right.
All right, looks like the next one's to you also, but I think you've largely answered it. Do you want to add anything to that, Meredith?
Yeah, I don't think I can add too much more than, as whenever you're doing long read sequencing, quality matters. So, handle samples gently and use as high quality a starting material as you can.
So great, Bob, I can take the next few here, if that's okay.
Yep.
So the next question is, I'm interested in the COVID Lineage app. Is it only available in base space? The answer to that question is no. We do have local versions available, and they're downloadable from support.illumina.com. If you have any issues with that, feel free to reach out to me. And this goes for everyone on the call in terms of anything Illumina related to my talk, jsmith6@illumina.com. I'd be happy to direct you to the right spot and also support your efforts there. The next question came in. There's an interest to survey broader amounts of pathogens from this morning's panel discussion. Can we expand, or can I expand on the capabilities of the Rpip assay and the specific system that these run on? So just to reorient, the Rpip assay is the respiratory pathogen ID and AMR panel that we offer. It's based on that hybridization technology using Illumina enrichment with RNA. So the targeted content of that panel is for bacteria, viruses, fungi, and AMR targets. So in terms of expanding on the capabilities, I just try to infer from this essentially what can we do with that? You're casting a broader net, and you're able to essentially go out and ID a broader set of pathogens that may be syndromic, for example. You're coming in with a pneumonia-like symptoms. It's not necessarily going to be viral, and you don't have to target it down. You can actually cast a wider net in order to detect them. The next part of that question, is there a specific system that these run on? The answer is no. It's scalable across all of the portfolio of Illumina sequencers in terms of the right application, or I should say the right run configuration to make sure that we're meeting your goals. That's something that myself and my counterpart on the East Coast would love to talk to you about in terms of the application for Rpip to be able to detect those particular samples and get you the right data at the right resolution. So the next question, how many unique barcode combinations are available to enable HTP, which is high throughput, sequencing of small microbial genomes? Short answer to that is that we do have 384 unique, so it's 4 sets of 96 that are available. But I do want to call out that the sequencers do matter in terms of how you actually utilize those, because some of the flow cells can actually be individually addressed in terms of the lanes. So to give you an example of that, on the NovaSeq, you're able to run up to 3,072 samples on a single run for SARS-CoV-2 on the high throughput instruments as well. I know that indexing can be something that, depending on which particular combinations you're looking at, there's other efficiencies that can be realized there. And I just encourage you to reach out to myself, and we can certainly get you on the right track there. So the next question, and this was at both of us, Meredith, I'll go ahead and take the first swatch at it. The question is, with your panel assays, is there any ability to also capture host genotype information simultaneously? So within the original COVID-seq assay utilizing the Arctic V3 primer set, there were human controls that were present there. In terms of the genotype information, this again gets back to essentially understanding the data that you're looking at and how to actually call that information. So for right now, the host pathogen informatics is definitely something that we're focusing on to bring that out, because that is really the next phase here of understanding not only what is the infection, but potential predisposition to those infections and how to turn that into clinically actionable information. So that's definitely something we're considering right now in terms of the host genotype information. Depending on the submitter of this question, I'd be happy to interact with you and understand what we could pull out from our existing tools, because I'm sure there's something there we can at least start down that
Yeah, so in terms of capturing both pathogen and host genotype information, I think one of the complexities with designing an application of that kind is always HIPAA requirements. So, for the COVID sequencing in particular, all of that data needs to be anonymized for reporting. And so, when we developed the kit, we didn't really consider including amplicons that would also amplify, human sequences specifically. But we do have some customers who are using the MIPS approach and then using that same sample to also look at, say, HLA in the human host. And obviously that's of huge interest, right? Trying to understand not only what were they sick with, but why do some people get very sick and some people get less sick? Why does the vaccine work great in some folks and not so great in other folks? So while you can't do it all in, from one box, there's absolutely ways that you can use the same sample to get both of those types of information.
And do you both have sort of recommended extraction methodology? And, again, it probably depends what you're after, and it would play into your last response there in particular, Meredith. But do you have prescribed sort of methodology for, to feed into these pipelines?
Yeah, so our customers have used a broad range of the RNA extraction kits that are out on the market. Off the top of my head, I know the QIAGEN kit has been used quite a lot. There's another from Thermo that's been used quite a lot. Again, since we are targeting a slightly longer amplicon, we do recommend that methods that are a bit gentler, while obviously still killing the virus, do tend to work better.
And I can jump in there too, Bob. In terms of the extraction methods and protocols, those are going to be key for anything downstream in terms of being dependent upon a high quality result. So absolutely, we've tested our assays internally, and we certainly have suggestions of what's worked, and these are actually built into the app notes as well. In terms of using one over the other, recommendations can be a strong word, but in terms of our capabilities to guide you in the right direction so you're not reinventing the wheel, we absolutely can help you with that. I think there was another one here. Meredith, I'll go ahead and take this as the initial as well, if that's okay. So I think the question is, where do PAC Bio and Illumina get feedback, or how are we alerted to emerging holes in the assay? That's a great question. It's something that I'm actually acutely interested in terms of keen conversations to have with members of this audience and the ones who weren't able to join us as well. I have these conversations all the time. As you can imagine, there's a trade-off and opportunity cost for every choice that's made in terms of these assays and what you're going after. We would love to hear in terms of the field about what you would like to see. And myself and my counterpart on the East Coast can always relay that back internally in terms of what makes sense. Are we casting too broad of a net? Should we have something that's more modular, more targeted? Does a specific assay base, for example, the enrichment versus the amplicon, make sense for a given sample stream? And then also data requirement that's coming out on the back end. So I would say just overutilize myself in that regard. If you see holes in these assays, we'd love to hear from you.
Yeah, same. So, with the COVID stuff, it's been super interesting. Honestly, there's like instantaneous feedback available on Twitter. And, the CDC holds a regular call, which has, folks from all over the country participating, giving feedback and updates on assays. And, I'm on those calls, I'm on Twitter, but then with stuff which maybe isn't quite so high profile, the best way to give that feedback, which, I agree with Jason, we're very keen to get, is to go in through our customer support email address. And I guarantee you everything that pertains to microbial sequencing will get in front of me through that mechanism. If it can't be sorted easily, or if it's just general feedback, it wends its way to me. And you're also welcome to email me directly or hit me up on Twitter. We're very keen to get your feedback on our products.
So Meredith, have you had to alter your MIPS at all? Have you had revisions to those to that panel in general, or has it held up through all the variations so far?
Yeah, amazingly, it is held up. So, when Omicron came out, we of course took all the probe sequences and threw them against the genome and, had a look at how we, were we gonna have to do some remediation or not? And what we found was that, we still had strong coverage in probes across the whole region. Because we have that dense tiling, it really puts a lot of, leeway for error. So even if some probes drop out, there's plenty of probes that still have zero mismatches just because of where they're sitting. So, obviously we'll continue to keep an eye on that, but so far, no, we've not had to do any revisions.
And Jason, with Omicron in particular, did the holes emerge first, or did you notice variants, or what does that look like, that process?
Yeah, it's definitely a bit of a chicken and egg problem where you don't know how to redesign a primer until you see the prevalence. I can say that, partnering tightly with the Arctic Consortia in terms of that redesign, it's something that we're acutely focused on when we see that rumbling start to come up in terms of how to enable the field on that. So as you can imagine too, in terms of turning, say, an oligo pool into a product, right? It could be too late by that time in terms of the need that's desired for detecting Omicron. And that was really the effort we took in terms of V4.1, was to enable the field based on, let's do some of the initial testing internally and then turn that around into an application technical note that can be utilized by the field. So it's always an arms race to some degree. And we've been living in this world even before the pandemic. We've been racing against this, I think, as a society. So, with NGS's capability to essentially build on backbones for the molecular assays, essentially we're just going to have to see and see what time tells, right, for these. But it's certain that we're very focused on making these things acutely available in the field as well. So I think there's one more question. Meredith, do you want to take that one? It's at both of us, but I've been stealing the first chair. I'll let you have first chair.
So the question is, I'm very interested in improving the high throughput extraction methods to balance the high throughput sequencing that we all enjoy. Are your companies thinking about a high throughput instrumentation for nucleic acid extraction?
You can imagine the automation partners that we have. We take our assays and build scripts for them along with those partners, which are available by the automation vendors themselves. But upfront from that, which say the COVID-seq assay, the extraction side of things, of course, now you've got another vendor in terms of their particular kit working on these platforms as well. So that's something that we have a full automation team, which I can proudly say has been built out quite a bit over the last 12 to 16 months here. So if you have questions about that in terms of your overall systems, or you want to know what sort of assays and scripts are available, certainly reach out to us and we'd be happy to direct you in the right spot.
I had a quick question. Has any of the variation outside of the spike protein been relevant in sort of pathogenicity and all? Or has it primarily been in the spike only?
It's a great question. I think, just overall, we talked about, or we've heard about in the media, this idea of a stealth variant, right? Where essentially you have the S gene target failure out of a qPCR assay. In terms of the ability and how that affects back to vaccines, this was covered in the panel content with Sharon and some of the other panelists there about sequencing is really going to give you the insight to be able to determine whether or not a variant that's on the rise has the ability to affect the efficacy of these vaccines. I don't have that data on hand in terms of other particular variants, in terms of their clinical efficacy and what that means for, say, their virulence. But I just think in general right now, we're just keeping ahead of this idea of being able to characterize the variants themselves. So I think that question remains to be answered.
Yeah, I think that's a really interesting question. And I'm going to take a stab at it, even though I'm not a virologist, and hopefully I'm not going to put my foot in my mouth in front of the virologists who may be in this audience. But, I think one of the really interesting things is that with the variants and the sequencing, a lot of the focus has been on those external protein domains. And those are certainly the most impactful in terms of vaccine escape and, immune escape in general, and also the efficacy with which the virus can enter into the host cell. But one of the interesting things that we've seen with Omicron, which I think is, kind of good news, bad news, is that while it was a lot more contagious, it was a lot less virulent also. And, Omicron had changes not only in those external facing proteins, but, in other regions as well. And I'm hopeful that once we get out of the constant state of emergency here with this pandemic, that, those kind of questions can be delved into a bit more, and we can use all of the data that has been gathered over the past year to understand, are there changes in the virus that happened that contributed to that lower virulence? And can we, kind of have ideas about which other proteins or domains or regions that we should be keeping an eye on, or that should be, flagged in terms of if we're going to do like surveillance in bats of all coronaviruses, what should we be looking at? And what should keep us up at night or not? And, I think these kinds of basic biology questions are going to be really exciting to dig into.
Right. Any last minute burning questions or any points that either of you want to make, in general terms about your technologies, or, COVID focused, any of the above? We've got about 2 minutes.
Yeah, I'll take 60 seconds here. Thanks, Bob. So I think just in general, first of all, thank you so much for the attention and your attendance at this meeting. It's very key for us to understand the two-way dynamic here in terms of our ability to present and speak with you on this panel side, but we're very interested in the next step. This is by no means the end of the conversation. And I think too, in terms of the pandemic and SARS-CoV-2 specifically, I was part of the Tiger team on SARS-CoV-1 when it came out, and that certainly didn't have the type of effect that we see with SARS-CoV-2 going into full-blown pandemic phase. But it was quite scary there for a few months. And in terms of the long nights that we were having in the pathogen informatics group, just to characterize that virus and potentially develop surveillance, tests and assays for that. I think in general, sequencing and thinking about functional analysis and in the future here that we're doing, I think we all agree that sequencing is really the tool that's going to open up continual doors for us to be able to answer these great biological questions like you just asked, Bob. So I can speak on myself, or on behalf of myself and everyone at Illumina. We thank you for your engagement and your capabilities to continue to push us to give you what you need in the field.
Yeah, I would echo that. I think as a scientist and a citizen of the planet, it's been very meaningful to me to be able to make a contribution to helping us all get through this pandemic. And I think one of the best things about it was the speed with which, everybody was contributing data and working together, both, in, corporate settings and academic settings and public health settings. And I think we all really, we're just blown away by how much we can accomplish when we work together and are exchanging all of that information. And I hope that that's something that, we can all remember as we move forward, that we can move faster together if we all have our eyes on the same goal, which I think is improving human health.
Well, great. Well, yeah, that spirit comes through just, in the interaction. We've got two competing technologies here, and, it's much more of a collaboration than a competition. And, that comes through, and, it did in the panel earlier today, and, in the interactions in this. So it's all your efforts are greatly appreciated. And thank you.
Thank you for having us.
So thanks again to the speakers. We'll clap. Unfortunately, we don't have a room full of people, but in June, so we'll just a reminder to please show up in June, face to face, and we can all sort of, talk face to face for a change. We'll have a little mask burning party, hopefully. Although I don't know that I want to burn mine anymore. We're gonna have to just save it.
Maybe just tuck it away. Just tuck it away.
Yeah, exactly. Just in case. And thanks to Sean for helping to chair this session. So just as a reminder, Sean, do you have any other final thoughts?
No, I don't. I think we just put a reminder out there for the remainder of the sessions for SFAF.
Yep. Yeah, so this evening, 7 p.m. Eastern time, we have 4 tech talks, 10x Genomics, SQL, DNA Script, and NanoString are going to talk to us from 7 to 8. And then at 8 o'clock, we have an open poster session. There's lots of great posters. Some have been up, some are new. So please revisit those if you've been already, because there have definitely been revisions and improvements to those. So with that, thank you all. And we'll hopefully see you at 7 tonight.
Great. Thank you.
Thank you.